Welcome to Medu. Today we are going to learn lipid digestion absorption. It is divided into two parts: digestion, absorption. Let's look about digestion first. So digestion takes place in stomach and small intestine. Stomach and small intestine. Now what are the lipids involved? For lipids we have cholesterol ester, TAG, as well as phospholipids. So these three are taking place in digestion, which is happening in stomach and small intestine. These three are also called the dietary lipids, where the dietary intake of an average human Indian is around 20 to 30 grams of lipids per day. Now let's quickly look at the digestion scheme. It's for TAG, it's for phospholipids and cholesterol ester. For cholesterol ester, the enzyme cholesterol esterase, as the name suggests, of cholesterol ester, hence cholesterol esterase, it breaks down cholesterol ester or the substrate to form FFA, free fatty acid, along with cholesterol molecule. Cholesterol is a very important molecule. We must understand that it's a substrate or a starting point for the synthesis of many molecules. Okay. For TAG we have three: LCFA, MCFA and SCFA. Long chain fatty acids, medium chain fatty acids, small chain fatty acids. For long chain, the digestion takes place in small intestine itself, while the enzyme required is steapsin, and the products formed are 2-FFA or 2 free fatty acids along with 2 monoacyl glycerol. Okay, 2 free fatty acids plus 2 monoacyl glycerol. For MCFA, again the site is small intestine, and the products formed are 3-FFA along with glycerol molecule. The enzyme required here is intestinal lipase. For SCFA, the digestion takes place in mouth or in stomach. Major part takes place in stomach, while a small fraction is also taking place in mouth when we ingest the food. When the food is ingested, the products formed are fatty acid or the free fatty acid along with diacyl glycerol. Free fatty acid, diacyl glycerol. For phospholipids, we have phospholipid A2. This is an enzyme which converts phospholipid into lysophospholipid along with a free fatty acid. This explains the digestion scheme of cholesterol ester, triacylglycerol, phospholipid, each converted respectively into cholesterol, MAG or DAG, monoacyl glycerol, diacyl glycerol, and the third, lysophospholipid. Now let's come to the absorption part, the lipid absorption part. Before proceeding, I need you to look at this diagram. This is a fat droplet. It got emulsified, or the emulsification of this fat droplet took place, and small fat tiny droplets formed. a micelle formation took place actually. They got emulsified by bile salts, bile acids, other emulsifying agents, lipase molecule or lipase enzyme, to form this micelle. Now this micelle has to enter to small intestine in order to get transported ultimately to reach the body, the whole body. This fat droplet contains all the dietary lipids, cholesterol ester, TAG, phospholipids, which are then emulsified to form micelles. Hence, this micelle also contains all the digested products. Out of all these things, the LCFA, the long chain fatty acids, are taken and transported to lacteals, while the remaining, the others, can freely go into blood capillary or can be easily absorbed by the blood capillary. Blood capillary doesn't like LCFA, while it definitely likes other molecules emulsified or involved in the micelle. So others can easily go into blood capillary while LCFA can't. So an indirect way is to go through lacteal method or go through lacteals. Now moving specifically to LCFA. This LCFA needs to go to lacteals in order to reach into the body. So what it has to do is, it has to take help of apoprotein. It's a protein molecule. It has to take the help of this apoprotein. However, it doesn't like LCFA. Even this apoprotein doesn't like, along with that blood capillary we discussed. However, it presents a condition that if it gets re-esterified, which we'll discuss later on, I have marked it, only then it can bind. So that's what happens. LCFA binds with apoprotein to form chylomicron. That's right, this chylomicron we have heard about. Now it can easily enter into lacteal passive diffusion through lacteals. It gets into lymphatic duct, all the lymphatic ducts, through which it goes into thoracic duct and then blood circulation. There are two anomalies here which we'll be discussing in a minute. 1, 2, 3. The third step I was talking about, it involves the re-esterification of LCFA molecules. They are re-esterified to form TAG again. Now let's look at the two anomalies we were talking about. The first anomaly is chyluria. I am talking about the clinics here. Second being chylothorax. Let's quickly look into these two. Now the two anomalies we are talking about. First one, chyluria. Second, chylothorax. What I mean is, first here, as lymphatic duct passes on to form thoracic duct, another accidental connection with urinary duct can take place, which leads to chylomicron in urine, hence chyluria. Chylothorax, same when thoracic duct gets connection with blood circulation, the normal blood circulation. Here too, an abnormal connection with pleural cavity can happen, which can lead to chylothorax. That means chylomicron in thoracic cavity, the pleural cavity, the lungs I am talking about here, in urea or urine. Let's look into clinics now. Clinics can be divided into two, as it's lipid digestion and absorption. So a digestion defect can happen and or defect of absorption. In digestion defect we have steatorrhea. Here the deficiency of steatocin enzyme or the pancreatic lipase we are talking about takes place. Since pancreatic lipase is absent, we can assume it's a pancreatic disease here. So in pancreatic disease we have steatorrhea, which leads to defect in digestion. Well, the anomaly is that unsplit fat is present in feces. This is the symptom. While in absorption defect we have some obstruction of bile duct or intestine removal. Obstruction of bile duct and intestinal removal. In both cases, split fat is found in feces. That's the difference between digestion defect and absorption defect. Unsplit fat in feces while split fat in feces. That's the clinics of it. So thank you for watching the video. Your watching this video really excites us to move forward, to improve, and is much appreciated, my friends. Thank you.